Okay, good evening and welcome. Just to respect everyone's time, I do want to start on time, and everyone, the other latecomers can trickle in as I give the introductions. But my name is Denise, and I am the representative of Pure Encapsulations here in Hong Kong. We also want to give a very warm welcome to Spectrum Innovations in the Philippines. They are the official distributor of Pure Encapsulations and also Douglas Laboratories, also practitioner brands. And we've been partnering with them to do a lot of education events for our practitioners. And so thank you everyone for logging in tonight and just taking part in this learning and discussion. Metabolic syndromes has been really a long-standing topic worldwide. Metabolic diseases contributes to billions of healthcare costs. And I know in Hong Kong, just if we look at obese and overweight, it accounts for about 55% of the population. And so clearly, and I think I was doing a bit of research in the Philippines, is 39, so slightly better than Hong Kong, but it's clear that we do have a long ways to go, and we need to approach this issue differently. And so tonight we have Dr. Denise Furness here, who will be going through a 12-week metabolic balance intervention with us. For those who have not yet had the pleasure of meeting and hearing Denise speak, Dr. Denise Furness is a functional geneticist, registered nutritionist, and registered exercise professional. She is a pioneer in the field of nutrigenomics and epigenetics. You'll definitely hear a lot of that in her talk. She began her career as a research scientist and has won national and international awards for her research and conference presentations. In 2012, she founded Your Genes and Nutrition and began applying her knowledge in clinical practice. Denise specializes in fertility, thyroid balance, and healthy aging. So before we start our session, I just want to ensure that there will be a time for Q&A, and you can feel free to submit your questions at any time in the Q&A box, and we can just go through them after at the end. And we will also be sending out the slides and the recording for those who are not able to join us for the whole talk tonight. So without further delay, I'll now pass the following time to Denise, the other Denise.
Thank you. Yes, the other Denise. So thank you so much. So firstly, I would like to disclose that I am an advisor for Pure Encapsulations. I think most of you know that if you've heard me speak before. So a big thank you to them for sponsoring this event, but also Denise and her family at Gyra Health. Did I pronounce that correctly? Is it Gyra? Yes.
Yes,
as well.
That's good pronunciation.
Yeah, I read it. I know what it is, but I don't think I've said it out loud before. But also Spectrum Innovations over at the Philippines. So we've done a few webinars this year, but we are really hoping to get over to Asia next year and do some work with you in person. So I'm going to touch quickly on metabolic syndrome. We're going to talk about this intervention protocol and then explore some case studies from this healthy aging pilot program. So this intervention protocol was actually put together to sort of reduce biological age. And if anyone heard the last talk, we talked a lot about epigenetics and, you know, the difference between chronological age and biological age. But what we found now that the data is coming in for those first 3 months is that almost everyone's cholesterol has improved. I mean, it's no surprise. We all know this is functional medicine practitioners, you know, with diet and lifestyle. But interestingly, there's been some epigenetic changes, which really, you know, take people from a high risk place of type 2 diabetes to a low risk place of diabetes. And that's quite exciting. And we'll get to that at the end of the talk. And as Denise said, any questions are welcome. I won't be too heavy on the genetics and epigenetics. I know it's not common testing in Hong Kong and the Philippines, though maybe as the years come and we do more work together. But if I do say something that you're not sure of, please ask. But I know that you're all very switched on, and most of the things I come up with, you actually end up asking me great questions and make me think. So moving on to metabolic syndrome, as we all know, this is a collection of conditions, including dyslipidemia, insulin resistance, and that excess abdominal fat. So really that, that adipose tissue around those visceral organs. As Denise was saying, this is a problem around the world, including the Asia Pacific region, and we are seeing an increase in frequency. And that's really what this table is showing here. So if we're looking on the bottom, down here on the horizontal axis, we can see the first lot of data is representing people in their 20s and 30s. Then we've got their 40s and 50s, and then, greater than 60. And what we're actually looking at is the prevalence of metabolic syndrome. So this yellow bar here, the one to the furthest on the left, this is representing sort of all the populations together. And what we can see is that at our 20s and 30s, about 20%, which is pretty significant for young people, do have or have been diagnosed with metabolic syndrome. This increases or doubles, in fact, to around 40% in our 40s and 50s, and gets close to 50% in those aged over 60 years. Now, if we do focus in here on what they show as non-Hispanic Asians, this is this bar here, this sort of in between the darker and the light blue, what we can actually see is that it is shown that there's a lower risk for metabolic syndrome in all age groups, 20s and 30s, 40s and 50s, and above 60 within Asians. But despite having a lower frequency or lower prevalence, you can see it increases with age. And actually, accelerated aging, or that's what we call when you sort of faster aging, but basically getting older, so advanced age, is the number one risk factor for chronic disorders, including metabolic syndrome, age-related conditions like Alzheimer's, cancers, things like that. So we want to stay as fit and healthy as we can, because then we have this sort of younger biological age. It doesn't matter what our chronological age is. If we're taking care of ourselves, we have this sort of younger cellular age, and we are less likely to develop things like metabolic syndrome. So talking about nutrition, we have already had these conversations. If you've been to previous talks, I talk a lot about diet. Today, I just want to keep it short and sweet in case you've been to those other talks, but the things we need to really focus on are water. So if someone's got metabolic syndrome or at risk for metabolic disorders and disease, the main drink they want to be having is water. They definitely don't want to be having drinks with sugar in them. Often when people are dehydrated, what do they want? They want sweet things. So they're going to turn to, sweeter foods or sweeter drinks. So being hydrated, drinking throughout the day can help with cravings, can help people keep, hydrated, helping with detoxification, all of these things. So some simple things I tell people to do, and actually even do myself, because I'm sure as a practitioner, you're like me, we've got all the knowledge, but do we act all the time, is to either have a jug. So for example, I've got a big glass bottle on my desk. If you know Voss, if you have Voss over there, I've got one of the big Voss bottles I've kept. I fill that up and try to have that twice a day. But people can just fill up a jug, have that on their desk or, somewhere in the kitchen. And when they come out, they can see how much they've been drinking. They know they're drinking throughout the day. And when it does come to older age, this is actually associated with an increased risk for dehydration due to decreases in thirst. So ensuring that people are drinking enough water. So aside from water, which is a really simple thing, and sometimes I know patients can get annoyed when you're just saying, I just want you to drink more water. They're wanting you to have, particularly if they're doing fancy, expensive testing, but it has to be one of the primary things that we advise. The other thing is protein and fiber with every meal. That is going to help people feel full. That is going to help with keeping their metabolism firing. If they're having if they're having enough protein, that's going to help with ensure that they're not going to, or less likely to be reducing their muscle mass. So sarcopenia is the word I'm looking for. So having enough protein, it's going to help them, even as we're aging too. And, with diabetes, it helps with wounds and healing and all sorts of things. So getting enough protein, in addition, the fiber. So the fiber is obviously going to help with gut health. We know that this has been associated with reducing LDL cholesterol, even improving balancing blood sugars. And this third point here I've got is actually, I had a slide, actually had a few slides in one of the talks I did for you a few years ago for the Philippines. And there was data showing that the average Filipino has an intake of 8 grams of fiber per day, which is only 30 to 40% of the recommended nutrient intake. I couldn't find data for Hong Kong on the average intake of fiber. Not sure if Denise or any of the practitioners there know that. But generally, here in Australia too, people don't get enough fiber, and that can come from, fruits and vegetables as well, as I mentioned in this study here. So this was published very recently in October 2023, and this was a randomized controlled trial in Hong Kong looking at a symbiotic, which was a probiotic and a prebiotic, as well as dietary changes. So the dietary intervention component was including enough vegetables and fruit per day to obtain 30 grams of fiber. So there really wasn't any regulations or sort of strict requirements in relation to other sort of food groups with the diet. It was specifically around ensuring that every day they had enough fruits and veggies to get that 30 grams of fiber. And then the symbiotic contained Lactobacillus acidophilus, so this species here, and Bifidobacterium lactis, as well as the prebiotic. So I've got the results here, but if you can see my mouse, I know there's a lot of information on this slide, but as I said, it literally was a dietary intervention. So 8 weeks with consumption of fruits and vegetables. And as I said, those fruits and vegetables, there was tracking and information to help them understand how they could get the 30 grams of fiber and that particular supplement. And what they found is that there was decreases in fasting glucose. And remember, this is only in 8 weeks. Improved insulin, their HOMA score, triglycerides went down, and there was an increase in HDL. The DSG is actually referring to diet and the symbiotic group. So those that actually, because they actually looked at diet alone and then the diet as well as the supplement. And what they showed is that when they had the diet and the supplement, it also lowered certain species of bacteria or microbes in the gut, which were linked with BMI, fat mass, and trunk mass. So this study suggested that increasing fiber from fruits and vegetables combined with this supplement is more effective than either alone. Now, the reality is we have tons of information around increasing, vegetables and fruits in relation to most chronic conditions and even reducing all-cause mortality. So people that eat 5 or more serves of vegetables per day generally have, better outcomes when it comes to age-related conditions and reducing risk of chronic disorders. So I've mentioned about protein and fiber. In our particular study, I did not recommend a specific protein supplement. I wanted people to focus on food. However, we did have fairly high recommendations around protein. I'm going to show that to you when we get to the slides with the protocol. So it was 1.6 to 2 grams of protein per kilo of body weight. There was also an expectation, actually my co-investigator was an exercise physiologist, so there was a lot of information around strength training as well. But if people were struggling to achieve those levels, we definitely suggested a protein powder. I personally take protein powder after I've trained. So the important thing is, getting a really good quality protein powder. Whey protein, if they're fine with dairy, or there are plant-based proteins. But obviously the whey tends to be better in the sense that studies show that this is, you know, more biologically sort of valuable in the sense it's just absorbed better. And when it comes to the protein powders, as you would all know, but just to reinforce, you know, sometimes if our patients or clients think they need protein powder, they might just go and grab something from the supermarket, but, you know, they need to understand that they don't want to have those additives, flavors, sweeteners. So if it is going to have a sweetener, something like a stevia or a monk fruit, rather than some of these artificial sweeteners that are out there. So when it comes to fiber, if people do need to boost their fiber, I tend to suggest psyllium husk, but lots of people struggle with psyllium husk if they've got dysbiosis or gut issues. It can be a bit harsh. They might get bloating, a bit of pain, wind. So the other is the sun fiber. So this was actually produced by Monash University here in Australia, specifically for people that were sensitive to FODMAPs or needed to be on a low FODMAP diet or have IBS, IBD, things like that. So inflammatory bowel conditions. And this is a very low reactive fiber that is still very good for the gut and less likely to cause those symptoms. So if someone does need fiber, as I said, I tend to sort of think of things like psyllium husk, but if they can't tolerate that, there is obviously the Pure Encapsulations product. So the Pure Lean that's actually got the sun fiber as well as some of these other prebiotics, like such as these oligosaccharides, as well as prune and apple pectin in it as well. So the other important thing when we're thinking about metabolic health and reducing risk for metabolic disease, or if someone has actually been diagnosed, aside from drink plenty of water and have protein and fiber with every meal, we definitely want to think about meal timing. So fasting has been shown to have lots of benefits, particularly around, you know, lipids, you know, blood sugars, weight loss, you know, even hormones. But the important thing to note is that this caloric restriction, these benefits that come that are even associated with extended lifespan or extended health span, when people are not malnourished. So it's calorie restriction without, you know, or ensuring that they're getting enough nutrients, and not doing this when they're already deficient. So we don't want people, this is probably not the best picture to use, but we don't want them to feel like they're being starved. If they're having lots of cravings, if they're, you know, if it's causing mood swings, things like that, we might not necessarily want to jump into, you know, fasting. And I actually don't do any extended fasts. I more do time restricted eating. For women, it tends to be sort of a 14-hour fast. For men, it could be up to 16 hours. However, in this study, we ended up having 60 people. Not all of them were my clients. So we did a podcast with my co-investigator, and we had actually over 300 people inquire. But when, you know, paying for the test and everything that was involved, we had about 60 sign up. So we suggested a 12-hour fast minimum, but women, if they wanted to, 12 to 14. And in some cases, we had some men that were already doing 16-hour fasts. So we were fine with them sticking with that. But as I said, if we are introducing these things, we want to make sure that people are not nutrient deprived, and perhaps before jumping into some of these things, making sure they've got enough minerals. You know, if they're low in magnesium, they might crave chocolate, or just, you know, working with them first so that they are set up for success. So when it comes to the protocol, they were told to drink filtered water throughout the day. So here is the amount. More if they're training or using the sauna, you know, if you're in the Philippines or Hong Kong, or even where I am, actually it's really hot today, Sunshine Coast, you know, if it's humid and hot all the time, you may, or you likely want to increase this. We advise starting the day with a hydrating drink, so that being water or a herbal tea before coffee. Minimum of 12-hour fast, as I just said, eating organic where possible, or looking for spray-free foods or wild-caught meats and produce, aiming to get most of their carbohydrates from vegetables, legumes, nuts and seeds, and some fruits, so more so these than the grains. Having their last meal approximately 3 hours before bed, avoiding snacks. Obviously avoiding sugars, highly processed foods, refined grains, aside from twice cooked starch. So we did, if people, I generally suggest brown rice, but if someone wants to have white rice, at least cooking that twice, and then that increases that sort of resistant starch. So they're getting increased fiber. It's actually less, you're going to absorb less of the calories, but actually then feeding all the gut bugs, and which is really good for short chain fatty acids and things like that. Aim for a maximum of 2 coffees, limit alcohol to twice a week. Now, ideally, it would be great if everyone just stopped alcohol altogether. But I did promise everyone that the protocol will be something they could stick to. And also, I think just from a long term, we don't necessarily want people to make drastic changes and then just fall off the wagon. So we're looking for something that they can do and they can stick with even beyond the study. Minimizing plastics and plastic food containers. That shouldn't say plastic food. Minimize plastic food containers and drink containers. And our first couple of sessions, when we started at the beginning of the year, we did provide education around endocrine disruptors, parabens, phthalates, plastics, things that have been linked with type 2 diabetes, metabolic disorders, PCOS, things like that. So we suggested or told them that they should aim for 6 serves of veggies per day, including 2 serves of cruciferous, 2 serves of leafy greens, and 2 serves of coloured. So I say leafy greens, but this could include, things like broccoli too. But if they put the broccoli in the greens, it can't be greens and cruciferous. It's the greens, and then the cruciferous would need to be, their cauliflower and, Brussels sprouts or whatever it may be. And then the coloured, which is really important for getting all those polyphenols, so including, purples, oranges, reds. I have to say, since doing this study, I eat a lot more beetroot. And even I've noticed a change in my shopping because I'm trying to get, 2 serves at least of coloured veg in per day. Even just when I do the shopping now, we're getting a lot more sort of coloured things. As I mentioned, 1.6 to 2 grams of protein split over the day. Initially, I was doing this myself too, and people had the option to track their food. I have a system where they can go in and log their food. Some people were struggling to get that. Even myself, like in the evening, I might be, well, I'm just going to have, some nut butter on an apple or hummus on a celery stick or something just to bump up my protein a little. I tend to only have a protein shake, after a strength training session, but on some days I was adding in a protein shake as well. Now that the study is coming to an end, I did say to people long term, they don't need to stick with this. Part of this study was trying to actually gain some muscle mass as well. So as I said, it was a healthy aging study, but what we found is these big improvements in metabolic health, as I said, cholesterol and diabetes risk. A maximum 4 serves of whole grains. So obviously, one cup of rice, cooked rice would be 2 serves. So that's 2 serves. And again, that's maximum, unless they had a heavy training load. 2 serves of nuts or seeds, things like pepitas are great. They've got like zinc and minerals and fruit. Particularly sort of the fruits like berries and things like that can be very, very good. Fermented food, 1 serve a day, healthy fats, and we said use herbs liberally, but that's been chopped off the bottom. So lots of education about the benefits of adding herbs to foods. They were advised to aim for 7 hours sleep most nights, including some breathing exercises, and aim to move 30 minutes a day. And there was an app. So the person who did the study with me, Paul Taylor, is actually an exercise physiologist. And we provide a lot of it, well, he's particularly provide a lot of information around sort of exercise movement snacks. I'm not sure if that's a term that you are familiar with yet. It is becoming more popular, but with the 30 minutes, they didn't have to do 30 minutes all at once. It could be, 3 10-minute things, or just in between. So for example, I'm standing. One good thing since the study is I'm standing more, or even just in between sessions, just doing little things. So movement snacks, strength training twice, preferably 3 times a week. Also some information around, mindfulness. And it wasn't part of the initial protocol, but something that Paul really wanted in there, and we put it in the app, was cold showers, and most people responded really well to that. So the app actually had a ritual board, and you could tick off every day, you know, I've had my supplements, I got my sleep, I've had my 6 serves of veggies, I've had my cold shower, you know, I've done my exercise. So the app was really great. We only had that for the first 3 months. Denise has actually done the test around biological aging, but we didn't have the app for the second 6 months. So when it comes to supplements, we included I put this together, you know, a B complex. So you might be familiar with this one if you're using Pure. So right here on my desk, but yeah, that's the B complex plus. I use resveratrol extra that had 100 milligrams of trans resveratrol. And I am going to give you some more information about resveratrol. I don't have the time, you know, within 45 minutes to kind of go through each supplement. I mean, with the bees, you know, the reason that that's in the protocol is, one, we know bees, you know, help with, you know, managing or helping, you know, balance homocysteine, keeping homocysteine within a healthy range. You know, high homocysteine is linked with cardiovascular health conditions as well as other things. In addition, the bees are really important for helping, you know, manage stress. So, you know, metabolizing our catecholamines. If there's any, you know, new practitioners or someone that doesn't understand that link, you know, when we have an enzyme COMT, also the gene catechol-O-methyltransferase, or COMT, it's needed to break down our catecholamines, including adrenaline, and it is a methyltransferase. So any methylation activities require our B vitamins, and in this case, it's got methyl, folate, it's got the methylcobalamin, so supporting methylation. So really keeping that homocysteine in check, supporting our, you know, getting rid of, you know, stress hormones, all that side of things, as well as, you know, mitochondrial support as well. The resveratrol, as I said, I'm going to go into a bit of detail. So the ubiquinol was there. So with ubiquinol, we know that ubiquinol decreases with age, a bit like things like glutathione. So by the age of 65, we tend to have, you know, half as much as we did when we were 25. And interestingly, I measured my CoQ10 levels and I was deficient before the study. I couldn't believe it. I'm only 45. But this is really important too for heart health, you know, thinking about cardiovascular health. And then we had a supplement in Australia. We used a supplement with nicotinamide riboside with some ATP support, sort of meaning magnesium, carnitine, things like that. Whereas those in Hong Kong and in the US, the UK and Canada actually used a different product because we don't have it here in Australia, the ATP, which I think it's called ATP support, mitochondrial ATP support, and it has NMN. So both nicotinamide riboside and NMN are precursors for NAD. And we know that NAD is really important for, you know, mitochondrial health, healthy aging, you know, reducing risk for metabolic disorders as well. And, you know, energy at the end of the day, I'm going to be sharing, you know, a couple of cases that have shown improvements in, you know, lipids and diabetes risk, but I would say that every single person has said their energy is so much better since doing this study. And then we had the Liver GI. It's a supplement I really like. It's got a little bit of NAC, sulforaphane, curcumin, and things like that. So the liver support. So they were the 4 that were included. That is the healthy aging sort of protocol. The supplements are included. We did do some blood testing along with the biological age, the methylation epigenetic testing. And if people were low in vitamin D, we then advised vitamin D. You know, not everyone was. If anyone was low in iron, that was obviously addressed, any nutrient deficiencies. And we discussed omegas. So here in Australia, we tend to not have as many of the oily fish as you do. I think you all have a better diet than us when it comes to seafood. You're likely to get a few more omega-3s naturally. So some people, they might have already been on omega-3s. I was happy for them to continue, or if they weren't eating, you know, any oily fish, you know, didn't want to, then we would suggest omega-3s. And Some people have asked why didn't, particularly there's a lot of, studies with omega-3s improving cardiovascular health, inflammation, brain health, healthy aging. The reason it wasn't in the protocol, there was a number of things you could imagine as a practitioner. We want to have, a number of supplements. This was sort of getting it down to the smallest amount. Not everyone wants to take lots of supplements. And then these were if they needed to have them. So moving on to resveratrol. So the reason why I want to spend a little bit of time on resveratrol is because there does seem to be a little bit of controversy around this right now. And a few people have said to me, should, even during the study, should we stop having the resveratrol? So I believe it was on a podcast, or maybe it's sort of filtering through that biohacking community, that resveratrol actually isn't good in humans. Most of the studies are in cell culture and animal models, but in humans, it's not so good. And to an element of that is correct. A lot of the studies around things like NMN, resveratrol, there's a lot of mouse model, yeast model, cell culture work, but we do have quite a bit in humans, which is what I want to share with you. But the reason this came about, and I was trying to find the study to add it in, and I did email the person that sent it, but I didn't get it in time because I didn't read it in detail. It was sent to me a few months ago. I scanned it, but it was a relatively small study. And what it showed was that I believe it was sort of older men had resveratrol. And in the end, along with sort of other things, there was a lower VO2 max. And they thought resveratrol might be reducing VO2 max. Now, the reality is it does actually have some impacts on nitric oxide. There could be something there. But I don't think it's worth saying don't have resveratrol from one study with a very small population. There could have been other things going on. So I don't know if anyone knows that study. Feel free to mention it. Inform me if I'm not relaying that information correctly. But from that, and I think it being on some major podcasts of people that, people that joined this study are interested in these things, there's been a lot of concerns over resveratrol. So when it comes to resveratrol, it's found in two forms, naturally, the cis and trans. It's really important from a supplement perspective that you are taking the trans resveratrol. This is the form that has been associated with the greatest beneficial biological improvements and cellular responses. In relation to antioxidant activity, it's shown to be anti-inflammatory. It supports mitochondrial function. And as we can see here, really over the years, because we do have quite a lot of research on resveratrol, has shown from a molecular mechanism to turn on lots of things at a biochemical cellular level that are beneficial. So, thinking about genetics, so, with antioxidants, so we know our endogenous antioxidant system is very powerful. So yes, we can take antioxidants like, vitamin C. Vitamin C is great, particularly liposomal vitamin C, or, other antioxidants, but we also have these endogenous antioxidants. So enzymes, genes that code for antioxidant enzymes, things like superoxide dismutase, things like glutathione peroxidase, catalase. And what we know is that resveratrol, similar to the properties of sulforaphane and curcumin, all these things, they upregulate, they turn on these genes that act as really powerful antioxidants. So resveratrol has that same ability. It's actually an epigenetic thing. It goes in and sort of talks to our DNA and turns on these genes. So resveratrol supports our antioxidant defense system, similar to a lot of those other bioactive compounds that we use as functional medicine practitioners, because we know that it's actually having quite a powerful effect. As I said before, inhibition of NOx, so could affect nitric oxide. It prevents eNOS uncoupling. Lots of sort of mechanisms related to dampening the immune response and reducing inflammation, so shown to have an anti-inflammatory effect, and other things affecting signaling. I mean, there's some studies even suggesting that it could be beneficial in cancers and things like that. I mean, I wouldn't necessarily be jumping and saying people go and use this to treat, but there are a lot of studies showing that it has beneficial effects for our cellular health and, of course, the mitochondria, as I said before. But even though we have lots of cell culture studies and animal model studies, there are some conflicting studies, and that obviously, we see that in research. But the reasons why is because sometimes they're actually using the wrong form. So in this case, they might not be using the trans form. Actually, it wasn't my research, but my supervisor, so for 10 years, I was doing research at academic level with government laboratories, CSIRO, and the hospital and the university, in fertility. So my first 10 years was in fertility. Then I had autoimmune stuff. From that, moved into functional medicine, recovered myself, always interested in diet and health and exercise. And then, sort of moved into a lot more metabolic and even weight. Most people are really interested in trying to manage their weight and be as healthy as they can. And then the healthy aging, which has really been the last 5 years, a lot of the healthy aging. So I have ApoE, I've got the risk for Alzheimer's dementia, and my mum had early onset dementia. So now I do a lot in that space. But what I got to before I went on that tangent was that the first 10 years of my career, I was doing research in pregnancy health and fertility in relation to genetics. So methylation, MTHFR, other genes, diet, things that would influence fertility in men and women. But my supervisor was doing a lot with vitamin E. So I mainly did folate and B vitamins. He was looking at vitamin E in relation to preeclampsia. They didn't see an association, the study didn't come up with any significant findings. It was then later established they used the wrong form of vitamin E. So sometimes these things happen in research. So if someone is doing research with the cis form of resveratrol, there might be a published study and they say, well, it didn't show to have any benefit, but you need to look through and say, well, what was the dose? What was the form? If it is in cell culture, what was the tissue? What was the cell? Maybe that is the issue. So when we look at doses, there have been a wide range of doses given. We don't know what the cutoff dose is for toxicity, but generally studies above 2.5 grams, which I think is a lot. I never give that much. As you saw in the protocol, we're giving 100 milligrams, but doses above 2.5 milligrams tend to give gastrointestinal symptoms and loose bowels. There are some studies with even higher amounts than that, but that's pretty excessive. Though, mind you, there are a lot of people online. As you know, there's all these sort of people online talking about healthy aging that promote, 1 gram a day. I don't think we need that amount. When we start to look at studies linked with cardiovascular health, this is one looking at heart failure. This study was a randomized controlled trial with 60 people in 2020, and they used 100 milligrams daily for 3 months and showed that resveratrol, shown there as RES, resveratrol improved several parameters of heart function, including systolic and diastolic, cardiac biomarkers, and exercise tolerance. So then looking at studies in relation to lipid profiles and diabetes, we can see these have all been chosen as randomized. There's obviously a lot more cohort studies, but these are randomized controlled trials, and we've even got a meta-analysis down there. And what we can see is doses as low as 10 milligrams. Sorry, there's a bit of when I've pasted this in, but as low as 10 milligrams has been shown to have benefits around reducing LDL cholesterol, 250 milligrams linking with improved glycemic control. And I don't need to read every one here, but what you can see that these are published randomized controlled trials with doses, ranging between 10 to sort of, 500 milligrams showing benefits. And this meta-analysis down the bottom, 396 subjects showing that 100 all the way up to 3,000. So I think, probably too high. People are going to be having gastrointestinal symptoms. Didn't significantly affect triglycerides, but did improve HDL. And I think I've cut a bit off the bottom there, but there were improvements. But again, getting into levels that are possibly a bit high, possibly causing some gastrointestinal issues. And then, that could affect absorption of other nutrients if there's inflammation or they're passing things, through the body quickly, not absorbing all their nutrients. In relation to blood pressure, there's a lot. Now, interestingly, I did not ask people at the beginning of the study or the end to measure their blood pressure, and I wish I did, because we have one lady, she's a lawyer. She's been on blood pressure medication for over 10 years. She's been through a very stressful time. She hasn't necessarily been able to follow the protocol specifically, but she has been good with the supplements. She went to the doctor and her blood pressure was low, and she's actually gone off her medication. So that's only one person, out of 60, no one else at this stage. I don't know how many people are on blood pressure medication. I wish I had have asked more about blood pressure at the start and at the end. We might repeat this next year. We're looking at getting some funding and linking in with NIM, which is our biggest integrative medical practice. But that's fascinating in itself, for someone to come off their medication and said that it's been a really stressful time for her within her practice from a work aspect as well. But lots of studies, linking the benefits of resveratrol with blood pressure as well. So the reason I'm showing you all this is, one, I wanted to trawl through the literature myself, but also be a bit clearer. It made me actually more confident in knowing there's actually a lot more studies in humans than I realized. Being randomized control trials, they tend to be smaller, but we do have other cohort studies with much larger populations as well, suggesting, benefits. The other thing that's come out of the study, because the epigenetic test is a blood spot, people have reported that when they've done their finger prick to do the second test after the 3-month intervention, they've noticed that their blood is running a lot more. So, for example, when they did the blood spot, so the little cardboard, they're doing the blood spot onto the cardboard. The first time it was quite difficult, and it was actually the same for me as well, but the first time it was more difficult to get that blood, the amount of blood, whereas the second time, the blood was just sort of flowing. So that made me check as well. And, I know, obviously, I think we all do, that omega-3s, there's a contraindication with having, blood thinners, but resveratrol has also been shown to be associated with reduced and slowed blood clotting. And taking this along with medications, we need to be mindful, that should say of, not PF, excuse me, but mindful of the interactions with these blood thinning medications. So moving on to some of the results. So this is actually our youngest person in the study. So she's only in her late 30s. And she's in the US. She's actually a dietitian herself, and uses a different reference range. I think you in Hong Kong and the Philippines use the same reference ranges as we do in Australia. So we're the millimole per litre, and nanomole, no, millimole. We're in the millimole per litre. So here we're looking at, milligrams per deciliter. But what we can see is that she was high in 2022. And again, at the end. So this person, I've actually looked at her genetics, her DNA as well. She has a very strong family history of cardiovascular disease and, heart attacks, heart attacks at a young age in her family history. And genetically, she's got a number of SNPs or genetic variations that increase her risk for cardiovascular disorders, and having higher LDL and having cholesterol. So we do know there's a genetic component, and often that can be harder to override. So this is only showing 2022, but she's reported that for years her cholesterol has been high, and she's not on medication, but has been trying to get it down. So this is what it was at the start of the study. We can see her LDL cholesterol is high. Her triglycerides, and I'm sure you probably think the same as me, even though the LDL is high, and sometimes this can be alarming for some practitioners. If the triglycerides are okay, if the HDL is good, I think it's, less of an issue. And in some cases, I haven't with anyone in this study, if there are concerns around, LDL and high cholesterol, and perhaps their GP's talking to them about going on statins or things like that, I tend to get a lipid subfraction so that we can actually look at the particle size of the LDLs, because sometimes people have got high LDL, but it's those lighter, fluffier, those, more buoyant LDL particles. So when you actually get the subfractions, we can then divide it up and find out, well, are they really at risk? If they are those small, more dense LDLs that are likely to get into the blood vessels and cause damage, then it's obviously more of a concern. And then, the other thing I request, I don't know if it's common there, but looking at a calcium score before people are jumping to medications, finding out if they really are at risk. So in addition to doing the bloods, obviously actually a big part of this study was using epigenetic testing. So we know that methylation, these methyl groups that sit on the DNA, this is the epigenetic mark we know the most about, is significantly linked with aging. So there are certain changes that can occur amongst these methylation patterns on our DNA. And in some people, certain changes will occur, and that's associated with what we call accelerated aging, increased risk of metabolic disorder and chronic conditions. And there'll be other people that don't have these changes. What we see in this person, as I said, we only had about 6 people out of the 60 that were at high risk of diabetes. And in this case, this person, as I said, this female who was, relatively young, she was doing marathons, quite fit, eating relatively well. She did drink alcohol. She didn't eat perfectly, but, not overweight. But as I said, the higher cholesterol, and she was a little bit surprised to see this epigenetic risk factor around type 2 diabetes. But what we found at the end of the intervention, so these are her results 12 weeks later, is that actually her methylation pattern had changed on these genes, and she went from being at a high risk to low risk of diabetes. And the reason I wanted to share this is because so this test is relatively new in Australia. It's a lot more popular in the US. I think functional medicine doctors have been using it there for a few years. I think Denise Tam might be one of the only ones to have done it in Hong Kong. And when it comes to the Philippines, I know there's a lot of practitioners that are very much into this stuff. Whether you're doing it in the Philippines yet, I'm not sure. But I spoke with the founder and asked him about the diabetes, because I was a little bit shocked that some of the people in the study were at high risk of type 2 diabetes, particularly because most of them coming into the study, they're volunteered to be in the study, they've paid for the test, they have a genuine interest in health. A lot of them were already taking care of themselves. And what I was told is that this particular marker is said to be robust for, 7-year risk. So if we see that this is low risk, they're fairly confident saying, well, for the next, 7 years, the chances of you getting diabetes is quite low. And when I asked, could it change in 3 months, in this 12-week intervention, the feedback I got was that it would be unlikely in such a short amount of time to reverse these epigenetic changes linking with risk of diabetes. So at that time, his name is Ryan. He said that the only thing that could change these risks within this amount of time were medications. So in saying that, we don't have a lot of studies on this yet. It's pretty new. But he said, yeah, aside from medication, they haven't seen epigenetic changes occur in such a short amount of time. But we obviously did. And so this is a second case. This is someone, a male in his late 40s. So we can see here his fasting glucose. Sorry, it's on an angle. These were scanned. When we look at his fasting glucose, it is within range. So the range is 3 to 5.4, millimole per litre. But it's still at the upper end. And when I went through this person's history, so I met with everyone at the start and I reviewed their results. What was interesting in this case is this person was, much stricter with his diet than the person before, as in, almost, I would say, extreme or sort of obsessed, in the sense that he told me, he was already, for the last 10 years, has already been eating this many vegetables, doesn't have any, processed foods, refined sugars. He wakes up at 4 o'clock in the morning to do sprints. and he had been fasting. And I thought, well, this is a relatively high glucose. I mean, unless it's the glycogen, breaking down, releasing glucose into his system to compensate. He's been exercising. But yeah, I thought, well, we want to keep an eye on this being, I'm not sure if you have an optimal range, but I generally like to see this a little bit lower. And particularly in this case, it was unusual that it seemed high when his diet, exercise, and everything was so good. The other thing I spoke to him about was possible stress. We know that stress can increase glucose, so the liver making glucose, pumping that out to do with the stress response, but he reported not being stressed at all. Looking at his lipids, so his LDL cholesterol is 2.4. And again, not too bad. It's under 2.5 for the general population. But if someone's at high risk for cardiovascular disease or has a strong family history, we say less than 2. And this particular person too had a strong family history of cardiovascular disease, but more so actually the type 2 diabetes than cardiovascular, unlike the first one who was more so a strong family history of really high cholesterol. So telling you a little bit about this male who has had an extremely good diet for 10 years, said that he's not, overly stressed, exercises, gets up to do sprints, how many mornings a week at 4 a.m., does strength training, read books. He reads books like Elizabeth Blackburn's, telomeres, very, very interested in all of this stuff. He was mortified when he found out that he was at high risk of diabetes. And he said he was going to change it. And actually, his was the first results I saw. And he did actually make some changes. So his glucose came down from 5 to 4.3, but his phosphate, which was low at the start, actually came down a little bit lower, and one of his liver enzymes. So I wanted to check that he was eating enough. I thought perhaps, with the phosphate, is he getting enough nutrients? But he assures me that he was following the protocol and that he definitely, even prior to, was having a significant amount of vegetables and healthy food per day. He said, one of the things he said to me, which will always stand out, he said, every meal he looks at, he says, how can I make this more healthy? What can I do to improve the nutritional content of my food? So he takes, eating quite seriously. So in what he has done, though, which it's good that his glucose, and you'll see his cholesterol or LDL did come down, is that he did add in more meat. He wasn't having that much protein. He went for lean meat, actually kangaroo, which in certain parts of Australia is popular, not everywhere, but it's actually a really lean, it's grass-fed, being kangaroo, so it's not, it's not, farmed the way that a lot of these other produce, beef and, chicken and things like that. So it's generally wild caught. We know it eats grass. It's a lean meat. It's very red. It's high in iron. It's actually really nutritious. So he was having more kangaroo, wild caught fish, etc. So the LDL came down slightly, but what was really significant was how much of an improvement he had from an epigenetic standpoint. So when I asked him what he had done, because it is quite a big shift, he said that he took out all, even, honey. So he wasn't having any sugar, any refined carbohydrates, but he said he was having honey. He took out, he had less, some of the high GI fruit. So he still had fruit, but, less bananas, things like that. But he did say that he was having honey most days. I actually think honey's great. It's antimicrobial and, good for people if you can get local honey and they've got allergies. I did say to him, it's not about staying off these things forever, but he was the first one where I saw this was reversed. So since doing these slides, I've seen one other person's results. They have also reversed. And there's a couple of other people's that I haven't seen. I don't know if all of them, the whole 6, will go from high risk to low risk, but it's quite exciting considering that the founder of the company, now I did want to show you, is it not there? I thought I had some other results. I wanted to actually, I haven't got any of the healthy aging results in here, but just to give you an idea, We have, and our oldest person in the study is 82. So she actually came out younger biologically. She's been doing a lot. I've worked with her daughter and have for 10 years, but she reduced her biological age, by another 6 years. It was, amazing looking at some of these results. So she definitely was not at high risk for type 2 diabetes. She's actually an Asian lady. She runs every day. She lives with her daughter. She lives as 3 generations all in one house, but she cooks for the whole family. But yeah, 82 and coming in much younger. But yeah, she definitely didn't have the risk factor for type 2 diabetes. Interestingly, though, her LDL cholesterol is high, and it didn't change much. So as I said, most people's lipid profile has improved, but not everyone. So I just want to mention, I've talked about epigenetics, and I didn't give you a lot of the foundations around epigenetics at the start of the talk. I really just wanted to focus on the metabolic side, the improvements in this diabetes risk. But if you are relatively new to epigenetics, we do have substantial evidence, and we know that it's epigenetics that really does play a role in the development of chronic conditions, including metabolic disorders. So these things that sit on the DNA literally impact genes being turned off and on, and they can either intensify susceptibility or can directly cause the expression or the development of metabolic disease. So when we talk about susceptibility, so for example, if someone is susceptible to type 2 diabetes, they might have genetic variations within a gene called SLC30A8. That's actually a zinc transporter that gets zinc to the pancreas, and zinc is needed to make insulin. So some people have a genetic variation that means that zinc transporter doesn't work efficiently. They don't get zinc to the pancreas, and that can affect insulin. So that is one of the big risk factors, as well as TCF7L2. That's the other big genetic risk factor. I mean, it's polygenic. There's not one gene for type 2 diabetes. There's quite a few genes that increase risk for type 2 diabetes. But even if you've got the genes, it doesn't mean you'll definitely get type 2 diabetes. It's when you've got the genes and then the environmental things happen. So for example, someone has SLC3082 and then they've got low zinc as well. Well, that exacerbates the situation. Or there are some epigenetic changes because of stress, trauma, exposures to chemicals and toxins, or nutrient deficiencies. These are all the things that can affect epigenetics, and that then causes the person to move from that state of homeostasis into the development of disorder. So in certain circumstances, these epigenetic changes that I'm talking about can persist across multiple generations. So it's not just about the individual developing these disorders. So we know that, for example, with men too, people often think about, women, the one carrying the babies, obviously the mother's diet. We know a lot of that now, how much some of the work that I did. So I used to measure methyl groups on the mother's blood and the cord blood, and looking at these changes in epigenetics with mums and babies, as well as the genes and the diet. But with fathers, we know a lot more in the last few years about the impact that the father's diet has epigenetically, not just on him, but also his offspring. So it can cause changes in the germ cells, within the DNA in the sperm, and that can be passed on. So fathers that are overweight, that have metabolic disorders, or metabolic syndrome, are more likely to have a child that is overweight and develop chronic conditions as an adult as well. And as I mentioned, these epigenetic changes are the result of interactions between our environment and the genes that we inherit. So we can't change the genes we inherit, but we know we can change our environment. And we've shown that in 12 weeks, we can make some big shifts, particularly when it comes to type 2 diabetes, or even reducing, LDL in someone who has had high LDL for years, and done multiple things to try to bring that down. For those of you who are interested a little bit in that test, This is one of the original papers looking at these methylation changes on these genes, so the epigenetic marks linking with type 2 diabetes. And there's been a number of studies since then. And just to sort of finish off, talking about epigenetics and insulin resistance as well. So I've obviously shown this particular test that shows diabetes risk. People are either at high risk or low risk. But there are other epigenetic changes too that have been linked with type 2 diabetes. And a lot of pharmaceutical companies are doing research in this area now and actually trying to target medications, pharmaceuticals that will change methylation on DNA. We also know that metformin can reverse some of the epigenetic marks that have been shown in pre-diabetic or diet-induced obesity mouse models. And at the end of the day, metformin too, has actually been linked with extended health span. But the reality is, what does metformin do? It's not just helping regulate blood sugars. People's appetite is reduced. They eat less. They don't want to eat. So because they're eating less, that helps obviously with weight. That then helps with balancing blood sugars. So if we can just teach people about some time-restricted eating, in most cases, they don't have to do extensive fasting. Some people can if they can tolerate it. But if they're aging and getting older, we know that, fasting, particularly more than a day, you're going to start to eat into your muscle mass. And as we age, it becomes very, very difficult to put that muscle mass back on. So you need to think about what is the end goal here, because if you're losing that muscle mass, that can affect your metabolic rate, your strength. And it's not just about physical appearance. As we age, we want to still be strong. We want to be able to, get up off the floor, lift things, do things. We want to keep as much muscle as we can. So in summary, metabolic health can be significantly improved in 12 weeks. Using a diet, high in vegetables, fruits, and whole foods, as well as movement and key supplements, we have shown an improvement in cholesterol, even in those who I said is genetically predisposed. Because I think most of us as practitioners know, and I'm sure many of you have seen it, that we can do a lot with lipids, with diet and exercise. But in those that are genetically predisposed, sometimes, what I see, because I do look at a lot of genetically predisposed people, is it can be very hard to shift. So something in this protocol, I'm not saying it was the resveratrol on its own. I focused on that because I've been questioned about it, but maybe the combination of all of it together has been extremely beneficial for lipids. In addition, what we have shown is that we can move people from an epigenetically high risk state for diabetes to low risk. And this is really, I think, the first time that this is being captured. Now, whether functional medicine practitioners are seeing this in the US or not, but as I said, Ryan, who is the founder, suggested to me that it wasn't possible to see something in such a short amount of time. So yeah, it's quite exciting. We are getting all of the data in now. I hope to have the statistics and the final information by about January. And there's a possibility, depending on the priorities of Denise, Gyra Health and Spectrum Innovations, we may come over and talk about this healthy aging protocol next year when we've brought all the 3-month and 6-month data together. So I thank you so much for your time. If you have any questions or comments, I would love them. Any question, any comment is welcome.